So in this video, we are going to look at aseptic technique. This is activity 1-3. So first, let's start with aseptic technique. Aseptic technique is to transfer bacteria from one place to another without contaminating ourselves or the culture. So we want to make sure that we keep that culture pure, that when we do the transfer, that we are only transferring the bacteria that we want onto a sterile slant or plate or wherever we are inoculating. Now, the word inoculate is referring to introduce microbes into a new environment. So that would be like if we were to take a bacteria that's in one slant and transfer it to a new slant. We would inoculate that. Now, you can think of inoculation if you think about vaccines, for example, right? You might have heard the term inoculate in that context. And it's the same idea, right? Because in a vaccine is often going to be an injection of a weakened microbe or a portion of a microbe that is used to elicit an immune response, right? And so that is still introducing microbes into a new environment. So what we'll be doing in this experiment is that we will be transferring Citrobacter frundi and Staphylococcus epidermidis. And these are the two bacteria that will be used in the gram stain. And so normally in lab, what we would do is we would have you get in pairs. And in pairs, one partner would make the Citrobacter frundi slant, and the other partner would make the Staphylococcus epidermidis. And so when I demonstrate this, I'm only going to demonstrate this with one of the organisms. I'm not going to demonstrate it with both because the same idea applies for both slants. So first, what is a slant? Well, it's an agar tube. So here's our agar tube. And the media is at an angle called a slant. So that slant is going to give us an increased surface area to be able to culture bacteria without needing an entire plate. And so this would be what we call a slant. So what you would do in this is, one thing I need to talk about is the Bunsen burner. And when we light the Bunsen burner needs to have what we call an inner cone and an outer cone. The inner cone, this blue dark part in the middle, that blue inner cone is the hottest part of the flame. So when we go to sterilize our loop is our instrument that we're going to use to make the transfer. We need to place the loop in the inner cone. And the inner cone is going to make it the hottest part of the flame, which will allow us to sterilize the loop. Now, the other thing that we need to be aware of with the Bunsen burner is that there is this little piece here that rotates that is called a baffle. And the baffle controls the amount of gas that comes through the Bunsen burner. So what you can see is you can see tubing here, and that tubing is bringing gas to the Bunsen burner. When I light this, I like to open the baffle, meaning the vents are open and air can move through it. I open the baffle before I light it, and that usually works well to give me my inner cone. So when we do our aseptic technique, you need to think about some general tips, right? Because I want to make a transfer from one tube to another. I don't want to contaminate myself, right? I don't want to get that microbe on me. And I also don't want to transfer unwanted microbes as well. So one of the things that you're going to see is that when you're doing a transfer of microbes, you want to stand back from where you're doing the transfer, meaning you don't want to do the transfer right underneath where you're working, because then things from you could fall into the culture. So I always do this work out in front of me. And when I do my transfers, the sterile area, the clean field, is right around the Bunsen burner. And so you'll see that when I do this, that I'm working out in front of me and I'm doing my transfer near the Bunsen burner, because the Bunsen burner is going to be our sterile area, right? Because this flame is going to prevent or help prevent microbes from getting into where I'm doing the transfer. And so I am going to demonstrate how to do an aseptic technique transfer. So here are the items that would be in your drawer. And the first piece of equipment is going to be a needle. And the needle is, like it suggests, it's just a little point. It has no loop. We also have a loop, and notice that it has a little circle at the end. That is your loop. We have our immersion oil for when we use our 100x objective. We have our lens cleaner to clean our lenses after we're done with our microscope. We have our striker to create the spark to light the Bunsen burner. And so there you can see you have to kind of push to get that spark. We have our permanent marker to help us label our tubes. We have a very high-tech slide clip holder, which is basically going to be a clothespin. That's our Bunsen burner, and that would be your test tube rack. And so those are things that you would use throughout the semester. So in this video, we're going to simulate transferring from the yellow cap tube into the screw top with the black cap. And before I begin, I'm going to loosen the black cap to make it easier to take it off when I'm working. So what I need to do is I need to get ready with the Bunsen burner, and I'm going to open the baffle. And that is going to allow air to flow through so that I can get a better flame. I'm going to turn on the gas, and I listen for the gas to make a hissing sound. If it's hissing too much, I turn it down just so I can barely hear it. And once I get that amount of gas right, then I'm going to light it with the striker. So my flame is lit, and I have my nice inner cone. Remember that the inner cone is going to be the hottest part of the flame. So I'm going to take my loop, and I'm just going to manually straighten it so that the loop itself is straight. And I'm going to start by flaming the loop, starting with the base and working towards the tip. And I need it to get red hot. The reason that we want to go from the base to the tip is if I have liquid on the tip of my loop and I put that in first, I risk it splattering. So I start from the base and work towards the loop. Now I'm letting it cool by holding the loop close to where the flame is. That's my sterile field. I'm going to pick up my tube that has my bacteria. Again, this is a simulation. I hold the cap between my pinky and my ring finger. I flame the opening of the tube, and then keeping my loop hand as much as possible steady, I bring the tube to it to pick up the bacteria. I flame the tube and I put the cap back on. Then I set that down. I move it in my rack so that I know that I'm done with that tube. Now I'm going to pick up the tube to make the transfer. I put the cap between my pinky and my ring finger. Again, I unscrew it. That was the advantage of unscrewing it first. Flame the opening of the tube. And then I'm going to inoculate that new tube with the bacteria that's on my loop. Bring the loop out, flame the opening of the tube, put the cap back on, and I'm going to set it down. Now, notice that there's still bacteria on my loop, so I need to re-sterilize it by flaming at the base and again working towards the tip of the loop. And once I do that, then I'm ready to set my loop down. So now we need to talk about types of media that we might inoculate using our aseptic technique. And so the first type of media is going to be a broth, and a broth is going to be a liquid culture. Typically, if we are inoculating a broth, we will inoculate using a loop. And so when I showed you the contents of the drawer, one utensil that was in there is a loop. It has this little loop at the end. And the other one is a straight wire, which is what we call a needle. So to inoculate a broth, we would use a loop. A slant, here's our slant, and our slant is going to have agar. It's going to be a solid, and it's going to be inoculated using a zigzag inoculation along the slant, and that is usually achieved using a loop. There are a few exceptions where we might use a needle. For example, in our citrate slant, when we get to our Invic, you will see that we actually use a needle in that case because we don't want to transfer too much bacteria. But usually if we're talking about inoculating a slant, it's going to be using a loop. And then the last one in terms of tubes would be an auger deep. And an auger deep is going to be a semi-solid. So it's not as solid as a slant, but it's not as liquid as a broth. It's somewhere in between. And so because this is a semi-solid, typically the way that this is inoculated is we do what's called a stab inoculation, meaning we're going to use the needle, right? And the needle doesn't have a loop, it's just a straight wire. And we're going to use a needle and we're going to stab inoculate the auger deep. You'll see this when we get to our motility test and our oxidation fermentation test. And so these are the various types of media that you will see us inoculating throughout this course. And you want to kind of have an idea of what type of utensil should you use to inoculate each type. So a couple key points when working with our aseptic technique. The first is going to be where you're working relative to where you're standing. So when you're doing your work with your aseptic technique, you don't want your rack and your tubes or your plate or whatever else you're inoculating, you don't want them right underneath you. Because if you're working right underneath you, the chance of contaminants falling in is much higher. If I work out in front of me and if I work close to the flame, then I'm going to lessen the risk of contamination because I'm not hovering over the tubes that I'm trying to inoculate. So I always want to work out in front of you and I want to work close to my flame. My flame is my sterile field. The other key that you want to think about is that when you're doing an experiment where you're transferring from one tube to the other, to keep track of where you are, I like to move my tubes within the rack. So like if I've picked up bacteria from this tube, I'll move it backwards in the rack so that it's not next to the one that I'm still going to use. Moving it in the rack just makes it a lot easier to keep track of where you are. The other thing that we need to think about is how we hold our loop. So the way that you hold the loop is that you're going to hold it like you would a pencil, like if you were going to write with a pencil. You want to hold it towards the back of the loop, not too close to the end, because if you're too close to where the loop is, the higher the risk of contamination, as well as potentially burning yourself. The other thing about your loop, or if you're using a needle, depending on what experiment you're doing, you want to make sure that the loop, the wire, is going to be straight. Because if these are in a drawer, sometimes this wire gets bent and your loop is like off to the side, and it's not going to be very well for inoculating a tube. So we always want to kind of straighten this loop before we get started. So just to kind of review what you saw in the video in terms of aseptic technique. So I'm going to walk through here, and then I have another video where I actually demonstrate transferring bacteria from one tube to another. So step number 1, label a sterile auger slant with your name, the date, the media, and the organism name. Now, one of the things that you're going to see when I show you the video of doing an actual transfer, on our tubes, we use those glass tubes and they have a white part on them. We don't want to label the white part or the label will never come off. So we label on the glass directly. You want to hold the loop in your dominant hand like a pencil. You're going to flame the loop from the base to the tip. And so notice that you start here and you pull the loop back until you get to the tip. And again, that is going to ensure that you don't splatter any liquid that might be on the loop. Then you would pick up the culture slant in your free hand, meaning you'll pick up the tube that has the bacteria on it. Remove the culture tube with the little finger of your loop hand. So there are 2 ways that this can be done. Some people like to put it in the palm of their hand like is shown here. The way that I demonstrated in the video, I actually put my palm side up. So I put my hand up and I put the cap between my pinky and my ring finger. Either way will work. The idea is you don't ever want to take the cap off and set it on the desk. That could potentially contaminate the cap. So I don't ever put the cap down. I will hold the cap in my hand that has the loop. And by holding it in my loop hand, I still have some flexibility to manipulate and to move my loop to get into the tube itself. And so I want to make sure that I don't set the cap down. I also don't want to leave the cap off the tube for long periods of time. I only want to open it as long as I need it and then immediately put the cap back on. So I remove the cap, and for me, again, I do this between my pinky and my ring finger, and then I hold the tube at an angle with the agar surface facing upward, meaning that I want to be able to pick up bacteria that's on my slant. Then I'm going to flame the opening of the tube. So I want to flame the opening of the tube so that any bacteria that might be on the lip of that tube doesn't fall into my sample. So I just kind of quickly pass the opening of the tube through the flame. So I'm going to pass it through the flame, and then I'm going to move my tube and I'm going to hold it at an angle, and then I'm going to move the tube up to the loop. So I move my tube up towards the loop, and I use my loop to pick up some of the bacteria that's on the slant. So when I pick up the bacteria with the slant, on the slant, then I'm going to remove the tube. I'm going to pull the tube out. And before I put the cap back on, again, I'm going to pass the opening of the tube through the flame. And I want to do that before I recap it. So you want to get in the habit of every time you take a cap off, pass the tube through the flame. You also want to pass the tube through the flame before you recap it. So you're going to flame it before and after. So once this tube is capped, then I would take this tube that I just finished using and I would set it in my rack because I am now done with that. I've already picked up my bacteria on my loop. Now, when I do this with my loop, I want to make sure that I'm not flinging my loop all over. I want to keep that loop hand as steady as possible to try and minimize bacteria from going all over. So I keep my loop hand as steady as I possibly can. Now, I have set down this tube that I picked up the bacteria from. Now I'm going to grab the tube that I want to inoculate, meaning the sterile tube that does not have any bacteria on it. So again, I'm going to pick up the tube and I'm going to put the cap between my pinky and my ring finger. And then I'm going to take my tube that I'm going to inoculate. And because I just took the cap off, I'm going to pass it through the flame. Again, I'm going to keep my loop hand steady and I'm going to bring the tube up to my loop. So very similar idea for how I picked up the bacteria is also how I'm going to inoculate the new tube. So I flame the opening of the tube, then I take my loop, keep my loop hand steady, bring the tube up to it. I want to hold my tube at an angle with the agar surface, the slant facing up. When I do this, when I inoculate this, I'm going to inoculate using a zigzag slant, a zigzag motion. So you're going to see this in a second, but I'm going to do the zigzag up to the top. When I do this, I don't want to pick up, sometimes there's condensation or water that's in the bottom of the slant. I just ignore that. I don't want to tip my tube so much that that liquid ends up on the slant, but I also want to make sure that when I'm inoculating this, that I don't go all the way to the bottom. I start a little bit above where the bottom is and I do my zigzag inoculation. And so this is my zigzag inoculation. I take my loop and I start at the base and I zigzag up to the top. Now, after I do my zigzag inoculation, again, keeping my loop hand steady, I'm going to take the tube out so that the loop and the tube are no longer together. I'm going to flame the opening of the tube. I'm going to put the cap on, and I'm going to then set that tube down. Now I need to flame sterilize my loop because I'm done with my experiment. I don't want to set down that loop that has bacteria on it. So just like before, I would hold my loop at an angle and I would flame it. Again, the last part to be flamed would be the loop itself because I don't want to splatter. So in this experiment, I would transfer bacteria onto the sterile slant. So this bacteria is on the slant growing, and I need to transfer from that tube onto the sterile tube. And so what I need to do is I need to start by labeling the tube. And I label it with my initials, the initials of the bacteria, the date, and my lab section. Now it's labeled, and I labeled on the glass and not the white part. Now I'm ready to start my transfer. Notice that my flame is already on. It has an inner blue cone for the hottest part of the flame. And I am going to make sure to straighten out my loop. And I'm going to flame the loop by starting at the base and working towards the tip. And so I'm getting the loop to be red hot. Now that my loop has been flamed, I let it cool down and I keep the loop steady and by the flame because again, the flame is my sterile field. So I want to make sure that I keep the loop over in the sterile field of view, and I'm going to pick up the tube that has the bacteria. And so when I pick up the bacteria, I'm going to put the cap between my pinky and ring finger, and I'm going to unscrew the cap because these are screw caps. I'm going to pass it through the flame, and then holding the tube at an angle, I bring the tube up to my loop, and my loop is going to touch where the bacteria is growing. And so it's picking up some of the bacteria, and I'm going to very steadily take my loop out, flame the opening of the tube, and put the cap back on. So now on my loop, I have my bacteria. I want to keep my loop hand steady. I need to inoculate the sterile tube. So I take the cap between my pinky and my ring finger, and I unscrew the cap, and I'm going to flame the opening of the tube, and then holding it at an angle with the slant facing up, I'm going to go towards the base of the slant, and I'm going to zigzag inoculate, working my way up. And so it's hard to see, but I'm just zigzagging, working my way up. Then I'm going to flame the opening of the tube and put the cap back on. Now again, am I done at this point? Answer is no. I need to flame my loop because it has bacteria on it. So I start again at the base and I work towards the tip of the loop. And so I need to sterilize that loop before I set it down. Now, the other thing that I have to do is I need to make sure that the cap is on properly before I put that in the incubator. And so what I do is I turn the cap all the way, I tighten it all the way, and then I give it about a half turn to unscrew it, and that makes the cap loose so air can still get in, but makes it so that the lid won't fall off. You must leave the cap loose when putting it in the incubator. And so now I'm ready to put it in the incubator.